So yesterday we were discussing about mitochondria, and at the end of the class we were discussing about these elementary particles or oxysomes, or we also call them as FOF1 subunits, right? Or we also call them as subunits of Fernandez-Moran. So we were discussing about their structure. Now you know each elementary particle is formed of three parts. FO part known as base piece, FO fraction it is known as base piece, acetalk, and F1 fraction that is known as the head, right? So this base piece, acetalk, and head, they form an elementary particle. So what is the function of these elementary particles? Why they are present in the mitochondria? As I told you yesterday, these elementary particles, they act as enzymes. They act as ATP synthesis. They act as ATP synthesis. They are involved in the formation of ATPs. So how do they form ATPs? You know, cellular respiration, it occurs in two phases. Cellular respiration occurs in two phases. One phase of cellular respiration is known as glycolysis. And another phase of cellular respiration is known as Krebs cycle. This glycolysis, as you know, it occurs in the cytoplasm. It occurs in the cytoplasm of cell. Glycolysis occurs in cytoplasm of cell. And in this process of glycolysis, glucose is converted into pyruvate. Glucose is converted into pyruvate. So the phase of cellular respiration, which occurs in cytoplasm of the cell, is known as glycolysis. Glyco means glucose, lysis means breakdown. So in this phase, there is breakdown of glucose. So in this phase, glucose is broken into pyruvate. And then this pyruvate, it enters into mitochondria. Pyruvate enters into mitochondria. It enters into the inner chamber of mitochondria. Pyruvate enters into inner chamber of mitochondria. In inner chamber of mitochondria, this pyruvate, it is subject to a series of changes in a cyclic manner. Those series of conversions in a cyclic manner, that is known as Krebs cycle. That is known as Krebs cycle. So in mitochondria, pyruvate is converted into one product, then it is converted into another product, then it is converted into another product, right? So it is conversion into different products in a series. And those changes are repeated in a cyclic manner. That is why we call those changes as Krebs cycle, right? In this Krebs cycle, coenzymes are reduced. NAD is reduced to NADH2, NADH plus H. Similarly, FAD is reduced to FADH plus H, right? So during this Krebs cycle, I am not going to discuss Krebs cycle here, right? I am just telling you what happens. In this Krebs cycle, as the products are being converted, one product into another, that into another, in this process of conversion of this pyruvate into different products, right? Coenzymes are being reduced. NAD, nicotinamide adenine dinucleotide, it is reduced to NADH2. FAD, flavin adenine dinucleotide, it is reduced to FADH2, right? So we say that these coenzymes are reduced because hydrogen is added to them. Then these reduced coenzymes, they release protons. And those protons are transported into outer chamber of mitochondria. These protons accumulate into outer chamber of mitochondria. You know, there is an electron transport chain, electron transport particles. There is electron transport chain that helps in the conversion, that helps in the carriage of these protons into outer chamber. As these protons accumulate in outer chamber, when these protons accumulate in outer chamber in high concentration, they create a proton gradient. Then because of high proton gradient, because of high concentration of proton, then these protons start flowing back into inner chamber. protons start flowing back into inner chamber. As these protons flow back into inner chamber, how they flow back into inner chamber? They flow back into inner chamber through these elementary particles, through this FO stalk and F1 piece. They flow back through them, right? And while they are flowing back into inner chamber, they release energy. They release energy. And that energy is utilized in conversion of ADP plus inorganic phosphate. ADP and inorganic phosphate are combined and ATPs are formed. ATPs are formed. So these electron transport particles or elementary particles, they are involved in the synthesis of ATP. So in the process of Krebs cycle, interconversion occurs. One substance, one compound is changed into another compound. In that process, coenzymes are reduced. NAD is reduced to NADH2, FAD is reduced to FADH2. Then as these reduced coenzymes are formed, then these reduced coenzymes, they release protons. Those protons are carried into outer chamber. These protons accumulate in high concentration in outer chamber. They create a proton gradient here. They create a proton gradient here. Due to this high proton gradient, then some of the protons start flowing back into inner chamber. They flow back into inner chamber through this elementary particle. Through this elementary particle. While they are passing through this elementary particle, they release a lot of energy. Protons release a lot of energy. And that energy is utilized in combination of ADP and inorganic phosphate. By the help of that energy, inorganic phosphate is combined with ADP. A phosphate bond is formed. And ADP plus inorganic phosphate, they are changed into ATP, right? So in this way, these elementary particles, they are the sites of ATP synthesis in mitochondria. That is why we call them as ATP synthesis. That is why we call them as ATP synthesis. So this is attributed as a whole to the function of mitochondria. So we as a whole say that mitochondria are the sites of ATP synthesis. In mitochondria, actually ATPs are synthesized by ETPs, electron transport particles, right? But we attribute this whole function to the mitochondria because Krebs cycle occurs in the inner chamber of mitochondria. In Krebs cycle, these reduced coenzymes are produced. Reduced coenzymes, they release protons. Protons are carried into outer chamber. From outer chamber, then protons flow back into inner chamber through elementary particles. They release energy. And by the help of that energy, ADP and inorganic phosphate is combined and ATPs are formed. So that is why they are known as powerhouses of the cell because they generate the ATPs, right? So that is the main function of mitochondria. So mitochondria are involved in the formation of ATPs, right? So that is all about the function, structure and function of mitochondria. I have studied cellular respiration. Not yet. Okay. After mitochondria, now we are going to discuss about another cell organ. That is endoplasmic reticulum. Endoplasmic reticulum. It is in short written as ER. So ER is the acronym for endoplasmic reticulum. Why it has got the name endoplasmic reticulum? Because it is present in the form of a network. Reticulum means a network. Reticulum means a network. It is present in the form of a network in the endoplasm. In the endoplasm of the cell. You know, the inner region of cytoplasm, which is present around the nucleus of the cell, that is known as endoplasm. The outer region of cytoplasm, which is lying just below the plasma membrane, which is transparent and homogeneous, that is known as exoplasm. So it is present in the endoplasm, around the nucleus. So that is why we call it as endoplasmic reticulum. Who discovered this endoplasmic reticulum? This endoplasmic reticulum, it was discovered by Porter, Claude, and Fulham in 1945. It was discovered by three scientists, Porter, Claude, and Fulham in 1945. And it was named as endoplasmic reticulum, named as ER by Porter in 1955. Porter gave it the name endoplasmic reticulum in 1955. Now you understand why it is known as endoplasmic reticulum. So first of all, it is present in the endoplasm. It is present in deep within the cytoplasm, right? Around the nucleus. So it is located in the endoplasm. That is why we say it as endoplasmic. There it is in the form of a network around the nucleus. So for that reason, it is known as endoplasmic reticulum, right? Okay. Now this endoplasmic reticulum, it is formed of three elements. Endoplasmic reticulum is formed of three elements. It is formed of one element known as cisternae. It is formed of another element known as tubules. And it is formed of another element known as vesicles. Vesicles, right? So it is formed of three elements, cisternae, tubules, and vesicles. So what are these cisternae? These cisternae, they are sac-like structures. Cisternae are sac-like structures which are found around the nucleus. This is the nucleus of the cell. This is the nucleus of the cell. They are found stacked around this nucleus. Cisternae are sac-like structures which are found stacked around this nucleus. This is one cisterna. This is another cisterna. This is another cisterna. This is another cisterna. So this is first layer of cisternae. Then they are present over another layer of cisternae. This is another layer of cisternae. They are stacked around the nucleus. So these are the cisternae which are present around the nucleus. And these cisternae, they are sac-like structures. They are sac-like structures. You can see in this diagram. They are stacked around the nucleus. These sacs, they are lying over each other around the nucleus. They have got continuity of lumen. They have got continuity of lumen. Continuity of lumen. These cisternae, they are sac-like structures. These sac-like structures, they are interconnected. So we say that they have got continuity of lumen. So they are interconnected with each other, and they are also interconnected with the nuclear membrane. So they have got continuity of lumen with each other, and they are also having continuity of lumen with the nuclear membrane, right? So there may be four to eight such sac-like structures around the nucleus. So these sac-like structures, which are stacked upon each other, having continuity of lumen with each other and with the nucleus, these sac-like structures are known as cisternae. They are known as cisternae of endoplasmic. Then we have got tubules. These tubules, they are tube-like structures. They are branching tube-like structures. Tubules are branching tube-like structures. These are branching tube-like structures, which may be joined with the plasma membrane. Which may be joined with the plasma membrane. So these branching tube-like structures, they are known as tubules. They are known as tubules. They may join with the plasma membrane, or they may join with the Golgi body. They may join with the plasma membrane, or they may be joined with the Golgi body. Right? This is the Golgi body. Golgi body. So second element of endoplasmic reticulum is tubules. What are these tubules? These are tube-like structures. We show some branching. They may be directly joined with the plasma membrane. They may directly get attached to plasma membrane. Or they may join with the Golgi body. Or they may join with the Golgi body. So this is the second element of endoplasmic reticulum. Then there are small fluid-filled vesicles. These small fluid-filled vesicles, these small fluid-filled spherical structures, they are known as the vesicles. These small fluid-filled spherical structures, which are covered by a membrane, they are known as vesicles. So these are the three elements of endoplasmic reticulum. These are the three elements of endoplasmic reticulum. So we say that this endoplasmic reticulum, it is formed of three elements. It is formed of sac-like structures, which are stacked upon each other, which are joined to each other, which have continuity of lumen with each other and with the nuclear membrane. Those sac-like structures are known as cisternae. Then we have got branching tube-like structures, which may be attached to plasma membrane, which may be joined with the plasma membrane, or which may get attached to Golgi body. Those branching tube-like structures are known as tubules. And then there are spherical, fluid-filled structures covered by a membrane known as vesicles. These are the three elements of endoplasmic reticulum. Now we classify this endoplasmic reticulum into three types. We classify this endoplasmic reticulum into three types. We classify endoplasmic reticulum into rough endoplasmic reticulum, SER, smooth endoplasmic reticulum, and another form that is known as SR, sarcoplasmic reticulum. So we classify endoplasmic reticulum into three types: RER, SER, and SR. RER, its full form is rough endoplasmic reticulum. Rough endoplasmic reticulum. SER, its full form is smooth endoplasmic reticulum. Smooth endoplasmic reticulum. And SR, its full form is sarcoplasmic reticulum. Sarcoplasmic reticulum. Right? So these are the three forms of endoplasmic reticulum. So which one of them is RER, which one of them is SER, and is sarcoplasmic reticulum visible here? So that portion of endoplasmic reticulum, which is studded with ribosome. The portion of endoplasmic reticulum which is studded with ribosome. The portion of endoplasmic reticulum which is studded with ribosome, which has ribosomes on it. That portion of endoplasmic reticulum is known as rough endoplasmic reticulum. The portion of endoplasmic reticulum which is having ribosomes on it is known as rough endoplasmic reticulum. Why we call it as a rough endoplasmic reticulum? Because these ribosomes, they are in the form of small granular structures. So when they are present on the external surface of endoplasmic reticulum, they give it a rough appearance. Ribosomes, they are in the form of small granules. These granules, when they are present on the external surface of endoplasmic reticulum, they make this endoplasmic reticulum rough in appearance. So that is why we call it as rough endoplasmic reticulum. So these cisternae, which are studded with a ribosome, these cisternae, which are studded with a ribosome, they form the RER. They form the rough endoplasmic reticulum. Those portions of endoplasmic reticulum which do not have ribosomes on it, those portions of endoplasmic reticulum which do not have ribosomes on it. For example, tubules. They constitute smooth endoplasmic reticulum, SER. Those portions of endoplasmic reticulum which do not have ribosomes on it. For example, tubules. They constitute smooth endoplasmic reticulum. Why we call it as smooth endoplasmic reticulum? Because its external surface is smooth. Ribosomes are not present. What made rough endoplasmic reticulum rough? The ribosomes, right? Here the ribosomes are absent. So external surface of the endoplasmic reticulum is smooth. That is why it is known as smooth endoplasmic reticulum, right? Then there are vesicles. These vesicles are also sometimes studded with ribosomes. If vesicles are studded with ribosomes, if vesicles are having ribosomes on them, then they become the part of rough endoplasmic reticulum. If the vesicles are without ribosomes, then they become the part of smooth endoplasmic reticulum. So you have to remember that those portions of the endoplasmic reticulum which have ribosomes on them, they constitute the rough endoplasmic reticulum. And that part of the endoplasmic reticulum which is not having ribosomes on it, that makes the smooth endoplasmic reticulum, right? Where we find rough endoplasmic reticulum? Are these two types, rough and smooth endoplasmic reticulum, together found in a cell? Or smooth and rough endoplasmic reticulum together found in a cell? Yes, they are found together in a cell, right? However, the content of rough endoplasmic reticulum or smooth endoplasmic reticulum, it varies according to the type of cell. There are some cells which are actively engaged in protein synthesis. There are some cells which are actively engaged in protein synthesis. Those cells which are involved in protein synthesis, they contain large proportion of rough endoplasmic reticulum. Those cells which are actively engaged in protein synthesis, they contain major portion of their endoplasmic reticulum in the form of rough endoplasmic reticulum. So the proportion of rough endoplasmic reticulum is higher in protein forming cells. Does that mean SCR is not present there? SCR is present there. Smooth endoplasmic reticulum is present in protein forming cells, but in low quantity. The proportion of SCR is very low as compared to the proportion of RER. Why RER is abundant in protein forming cells? Why RER is most abundant in protein forming cells? Because it contains ribosomes on it. You know, ribosomes are the protein factories of the cell. Ribosomes, they form polypeptide chains. Ribosomes release those polypeptide chains in the lumen of rough endoplasmic reticulum, where those polypeptide chains, they assume a secondary structure, a tertiary structure, and a quaternary structure to become functional proteins. We have discussed structure of protein. You know, first proteins get a primary structure. In the primary structure, they are in the form of a linear polypeptide chain. Then that polypeptide chain may become alpha helix, that may become a beta pleated sheet, that may become a triple helix. So that becomes their secondary structure. Then it may further get tertiary structure, then quaternary structure, right? So ribosomes, they synthesize polypeptide chains. Ribosomes are present on rough endoplasmic reticulum. They synthesize polypeptide chains. Then they release those polypeptide chains in the lumen of rough endoplasmic reticulum. And in the lumen of rough endoplasmic reticulum, those polypeptide chains, they assume a three-dimensional structure. So that is why rough endoplasmic reticulum is mostly found in protein forming cells. It is most abundant in protein forming cells. So which cells are actively engaged in protein synthesis? For example, we have got fibroblasts. Fibroblasts. These fibroblasts are connective tissue cells. Fibroblasts are connective tissue cells. They form collagen protein. They form collagen protein. They form that elastin protein. Elastin protein, right? So these are protein forming cells. So they are actively engaged in protein synthesis. So they have higher content of rough endoplasmic reticulum. They have higher proportion of rough endoplasmic reticulum. Similarly, there are some cells in the liver which synthesize proteins. Those cells of the liver which synthesize proteins, they also have got a larger proportion of rough endoplasmic reticulum. Similarly, we have got cells of pancreas, which actively produce enzymes. Enzymes are mostly formed from proteins. So cells of pancreas, which are actively secreting enzymes, they are also rich in rough endoplasmic reticulum. So all those cells which are engaged in protein synthesis, they will have a greater content of, they will have greater proportion of rough endoplasmic reticulum, because rough endoplasmic reticulum bears ribosomes on it, and it is actually ribosome which is forming the polypeptide chains. Then ribosome releases that polypeptide chain in the lumina of rough endoplasmic reticulum, where those polypeptide chains, they become bent, fold, and they assume the secondary structure, tertiary structure, or the quaternary structure. So that is the reason why rough endoplasmic reticulum is found in greater proportion in protein forming cells. Then where we find SCR, as I told you, they are both present together in a cell. But in protein forming cells, RER is in greater proportion. In lipid forming cells, SER, smooth endoplasmic reticulum, is in greater proportion. So SER, smooth endoplasmic reticulum, it is found in large quantities. SER is found in large quantities in lipid forming cells. In lipid forming cells. So it has got enzymes for the formation of lipids in it, right? Which cells are actually engaged in lipid forming cells? Lipid, which cells are actually engaged in lipid formation? For example, we have got adipose tissue, cells of adipose tissue, fat forming, fat synthesizing and fat forming tissue. In adipose tissue, the cells are rich in SCR, smooth endoplasmic reticulum. Similarly, we have got adrenal cortex, cells of adrenal cortex. Cells of adrenal cortex, they are rich in SCR. Similarly, we have got some liver cells which synthesize lipids. They are also rich in SCR. So SCR is in greater proportion in those cells which are actively engaged in lipid synthesis. For example, adipose tissue. Adipose tissue. For example, adipose tissue. Adipose tissue is actively involved in fat synthesis, lipid synthesis. So that is rich in, those cells are rich in SER, smooth endoplasmic reticulum. Adrenal cortex, it is forming the steroid hormones. The cells of adrenal cortex, they secrete steroid hormones, hormones formed from lipids. So that is why they are rich in smooth endoplasmic reticulum. Some liver cells, which produce the lipids, they are also rich in smooth endoplasmic reticulum. So now we come to know that both SCR and RER, they may be present together in a cell. But which one of them will be in greater proportion? In protein forming cells, RER will be in greater proportion, because protein cell forming cells will have most of the ribosomes in it. And those ribosomes are present on the surface of endoplasmic reticulum. So that makes it rough. So rough endoplasmic reticulum is abundant in protein forming cells. Whereas lipid forming cells, fat forming cells, they are rich in SCR. They do also have RER, but in them RER is in lesser proportion, and SCR is in greater proportion. So these cells, for example, adipose tissue cells, adrenal cortex tissue cells, liver cells, which synthesize fats, they are rich in SCR, smooth endoplasmic reticulum, because in this smooth endoplasmic reticulum, there are enzymes which are involved in fat synthesis, which are involved in steroid formation. That is why it is found in fat or lipid forming cell. Then we have got the third type of SR, known as sarcoplasmic reticulum. Where we find this sarcoplasmic reticulum? We will discuss now that. Sarcoplasmic reticulum is a modified endoplasmic reticulum. It is not found in all cells. It is found in some special cells. Sarcoplasmic reticulum, SR. Sarcoplasmic reticulum. It is found in skeletal muscle cells, particularly. Skeletal muscle cells. Sarcoplasmic reticulum is mostly found in muscle cells, particularly in skeletal muscle cells, right? You know, there is different terminology for a muscle cell. The plasma membrane of a muscle cell is known as plasma membrane of a common cell is known as plasma lemma. Plasma lemma. Plasma membrane of a common cell is known as plasma lemma. Cytoplasm of a common cell is known as cytoplasm. Right? But in muscle cells, the terminology is different. In muscle cells, the covering membrane of the cell, that is plasma membrane, it is known as sarcolemma. It is known as sarcolemma. The cytoplasm of a muscle cell is known as sarcoplasm. Is known as sarcoplasm. Similarly, endoplasmic reticulum of a muscle cell, endoplasmic reticulum of a muscle cell is known as sarcoplasmic reticulum. Is known as sarcoplasmic reticulum. So sarcoplasmic reticulum is the endoplasmic reticulum, which we find in the muscle cell. Since in the muscle cells the terminology is different, in the muscle cells we call plasma lemma as sarcolemma. In muscle cells we call cytoplasm as sarcoplasm. Similarly, in muscle cells we call endoplasmic reticulum as sarcoplasmic reticulum. Though it is different, although it is different from the rough endoplasmic reticulum and smooth endoplasmic reticulum of common cells. So we will discuss about sarcoplasmic reticulum inshallah tomorrow. We are running out of the time. Inshallah, we will discuss the sarcoplasmic reticulum tomorrow. Thank you for being with me.